All right, the beginning of chapter 10 in our book starts with sort of the story of how scientists got the idea of how DNA actually converts itself to protein. So the story begins with a problem, and Archibald Garrod noticed that a lot of kids were having this issue. Their urine was turning either dark brown or black once they actually peed, and this was causing some mental disorders in the children, and he noticed that this was very common among first cousins. So they gave this name alkaptonuria, but I don't really care about the name that much. Just sort of understand the focus of the story is that he sees this issue with cousins. We know how heredity works, and we know that things that are passed through families tend to be more recessive traits. That's one of the big issues with mating with people that are of close relations, is that recessive traits are going to go forward in the generations. So he figures, you know what, this must be some kind of recessive condition that's allowing this problem to happen in the children. And as he's looking at some of the body fluids, the blood, fluid out of the joints, inside every joint there's a capsule with fluid in it, and inside the urine, he keeps finding this stuff. It ends up being homogentistic acid. And he's like, well, if these people are having a problem and I'm finding homogentistic acid, then there must be something that people that aren't affected with alkanopturia, they must have some kind of enzyme that's breaking down the homogentistic acid that these kids don't. So he's figuring out that there must be some kind of enzyme that is what we call the wild allele, or the thing that would normally be present, since it's more common to not have this issue. And if you don't have that, you're not creating this allele. You have some kind of mutation going on. And he said that this whole idea was some kind of inborn error of metabolism. In 1958, eventually, we finally figured out that there was something called homogentistic acid oxidase, which hopefully you know that the ASE at the end of that name tells you that that's an enzyme. And that's the stuff that's going to take care of the acid inside of the person's body. And the actual mutation that he described before we even knew what this enzyme was that was causing the urine to be brown or black was first described in 1996. So he was way ahead of his time when he was doing this. And when homogentistic acid is breaks down those proteins, it leaves phenylalanine and tyrosine as byproducts if it's not broken down. So ideally, that homogentistic acid needs to be broken down by homogentistic acid oxidase. Well, there are other conditions that are similar to this. Most commonly is something called phenylketonuria, or PKU. One of the first things a doctor is going to do when you ever have a kid is they're going to prick the heel of the kid and take a little blood sample and try to figure out, is this infant positive for having this PKU disorder? Because if they are, you have to modify their diet, because if you don't modify the diet, then the baby, as it's developing and eating the specific proteins that are specifically found in the mother's milk, will lead to mental retardation, which ideally we try to avoid that kind of thing. So if they find PKU in the infant's blood, then they will modify things. If they don't find it in the blood, then your infant's going to be fine. So as we're looking through all of this story, we're starting to get the idea of how a protein works, how the DNA and protein relationship works. And this initial idea, this initial gene concept that was developed by scientists, is the idea that one gene equals one protein. And whatever's in the DNA is exactly what comes out in the protein. This is a very simplistic view, but it gave us a starting point as to how we understand the way that DNA is converted into protein. And basically what this idea says, if you just change the amino acid sequence, you're going to change a protein. Well, again, very simplistic view, something that we have to improve upon a little bit. One protein that scientists use to study really commonly is called hemoglobin. Hemoglobin is a really good protein to study because it has quaternary structure, where, if you remember, primary structure is held together with covalent peptide bonds. Secondary structure is hydrogen bonds in between R groups that cause either alpha helices or beta pleated sheets, and then tertiary structure and quaternary structure are both held together with possibly hydrogen bonds, ionic interactions, hydrophobic hydrophilic interactions, van der Waals, disulfide bridges. All of those things can help fold the protein into shape between the R groups of the different amino acids that's going to create the different structures. Hemoglobin particularly has 2 alpha helix chains and 2 beta pleated sheet chains, and those are held together through all of those bonding mechanisms. And one common disorder of people that affects the hemoglobin is something called sickle cell, where instead of the red blood cells being nice and round, they are shaped kind of like crescent moons, which is what a sickle looks like. The thing that, if you think about Death, the guy with the black robe, the skeleton face, and the stick with a curved metal part, that curved metal part is called the sickle. So that's sort of what the blood cells kind of look like, and that's how it got its name. Well, there's a lot of different alterations to the gene that we found in that create the hemoglobin for a person, but not all of those alterations change what the protein does. So this meant that just changing the amino acid sequence doesn't always change a protein because of all those special interactions that we talked about. If the interactions aren't changed because you substitute one amino acid for another one that has basically the same properties, well, then we're not sort of changing anything. So this sort of led to a little bit of a more advanced view is that one gene converts to one polypeptide, which, as we're going to learn very soon, is still not a perfect one, but you know what, it's a little better than one gene to one protein. Genes get expressed as phenotypes, and they get expressed through basically from RNAs working together to turn that DNA blueprint for our cells into working proteins. And that transition happens through 2 processes. The 2 processes where genes are expressed are called transcription and translation. Transcription is when DNA gets converted into RNA, mRNA to be specific, but we'll get to that in another slideshow. Translation is a conversion from RNA into the actual protein, or into that amino acid sequence that's going to be the eventual final protein. There's a lot of modification that goes along, and I'd like to say that it was just as simple as, oh, transcription, translation, but there is a lot of stuff going on into it. And the next couple slideshows are going to get us to that point. So hopefully you enjoyed this one. I will see you later.